All right. Well, thank you, Kevin. And thank you everyone for joining our session today on food allergens. And my name is Jason Haluka, and I am with the Kraft Heinz Company. It is my privilege today to help facilitate this session. And First, I wanted to introduce our speakers, but first just wanted to start off with a few housekeeping items, just for clarity. So we have three excellent speakers that will be presenting on a variety of topics related to new frontiers in food allergy testing and patient care. And what we're going to do is to run these sessions consecutively. And we would ask that if you have questions of the speakers to use the question box. So again, there's some instruction that was provided previously, and if you hadn't joined, but if you go to your little pop-up screen on the right, you should see a drop-down option to pop open a questions tab. You can include your question there and send it off. They come in directly to IFIN staff, and we'll be capturing those. And after the session, there'll be a short Q&A session. If we don't get to your question, we'll certainly have the speakers address. We'll compile all of them and have the speakers address those. There's also the option of adjusting the size of that box. So if you have difficulty, it's a little bit small. So if you could blow it up, that's helpful. So just for starters, I'm going to, and within that field, sorry, just one more thing. Within that box, there is an option as well. I'm not sure which tab it's in, maybe the dashboard or something like that, where there are files. And so you'll find full bios for all the speakers there, as well as the full program agenda. So just to get started here in our speaker session. So as I said, we're going to have three speakers, and the first speaker, it will be Dr. Stefano Luccioli. And Dr. Luccioli is currently the Food Allergen Program Coordinator at the Center for Food Safety and Applied Nutrition at the US FDA. He is also on the clinical teaching staff at Georgetown University Hospital in Washington, D.C., and he'll be providing a presentation on the current US food allergen regulatory landscape. Next, we'll hear from Dr. Bruce Roberts, who serves as the Chief Research Strategy and Innovation Officer at FARE, where he leads FARE's scientific research initiatives. And Dr. Roberts will be talking about the FARE Clinical Network and advancing new treatments, preventive therapies for food allergy patients. And finally, we'll hear from Dr. Paul Kearney, who is the CEO of Data Insights and also the strategic advisor for Allergenis. And his focus is now on breakthrough tests in food allergy and the application of data science to important clinical problems. And Dr. Kearney will be talking about opening the allergy black box, the path to risk-free challenge testing. So there's a little bit of a different aspect to food allergens. We've, IFINs and previously we've had sessions related to different aspects of food allergy. And in this one, we're bringing forward a more consumer-focused perspective. So I hope you'll enjoy the session. And once again, submit your questions as well as access the various handouts that are accessible in the fields. And without further ado, I think I will turn it over to Brianna and the presentations. Thank you.
Thank you, Jason. My talk today will be on the current food allergen regulatory landscape. I have no financial interest to disclose, but I do want to disclose that this talk is my talk and not representative of the Food and Drug Administration, since I will be talking about some issues regarding thresholds that are not established by the agency. My objectives for today's talk is to provide some background on some key regulatory activities involving food allergens and labeling, explain some gaps and challenges with avoiding allergen hazards in products, and then talk to you about some of the recent assessment and discussions around allergen thresholds, which is a very big topic in allergens these days. Just to present the argument, food allergies are now defined as IgE-mediated adverse responses to foods. In general, they are all immune responses, but we refer to food allergies as those that are IgE-mediated because they can lead to anaphylaxis and are potentially fatal. It involves foods and proteins that are commonly consumed in the diet. Prevalence in the US is not so accurate, but probably about 3 to 4% of the population. But some studies have reported children and adults 6 to 8% with a food allergy. So it seems to be on the rise. It's both genetic and environmental. There are promising treatments, but no cures. And so labeling and avoidance of the allergen hazard is key. Avoidance, though, remains difficult, and this leads to a lot of issues, not just with accidental reactions, but to decreased quality of life. And there are also economic costs as well. My center, the Center for Food Safety and Applied Nutrition, its mission is to ensure safe, sanitary, and honestly labeled food supply. And in regards to allergens, this is to make sure that products with allergens are properly labeled and identified by consumers so they avoid harm. We do so by a number of different regulatory activities. We implement food allergen labeling laws, develop policy and other guidances and educational efforts. We have a laboratory that develops methods to detect allergens in different food products. And we have a big enforcement and compliance division that does inspections and also manages recalls of undeclared products with undeclared allergens. We also have a science board that looks at different types of questions regarding novel food ingredients, health claims, health hazard evaluations, and also risk assessments, including thresholds. And we also have, through MedWatch online, consumers can report to us problems with FDA-regulated food products, and that's managed by the CFSAN Adverse Event Reporting System. It's important to realize that managing allergens is a much larger effort, and there are other centers within the FDA that have an important role with food allergens. Importantly, a lot of the recent treatment that you've heard about, especially Palforzia, which was approved, first ever approved medication for food allergy, was approved in February 2020. Allergen extracts and as well as allergens and vaccines, they are regulated by the Center for Biologics. Treatments such as epinephrine and other biologics that might be used in managing anaphylaxis are regulated in the Office of Drugs and Evaluation and Research, CDER. Diagnostic tests and genetically engineered salmon are in different centers as well. And it's important to realize that not all food products are regulated by the FDA. Distilled spirits, wine, beers are by our sister agencies, the TTB, meat, poultry by USDA, and pesticides by the EPA. But importantly, the principal laws that govern allergens is the Food Allergen and Labeling Consumer Protection Act of 2004. And this came into effect in 2006. And this defined what we call the 8 major food allergens listed here: peanut, milk, egg, soybean, wheat, fish, crustacean, shellfish, and tree nuts. We have a fairly broad list of tree nuts, which also includes coconuts. That's different from some other countries. But lo and behold, as of April of this past year, the FASTER Act was passed. And what this does is this will add sesame to this list. So now we will have 9 major food allergens, and this will come into effect on January 1st, 2023. We also have a draft guidance for industry in place that until then, instances where sesame may be found as a spice or flavor, we encourage that this presence be voluntarily disclosed. Another important requirement that the FASTER Act is that it requires the Department of Health and Human Services, which houses FDA, to issue a report to Congress. And so also to highlight the different types of activities going on with food allergens, what we know about the prevalence and severity of allergic reactions, food allergy diagnostics, which you might hear later today, some new promising diagnostics, the prevention and reduction of risks for food allergies, as well as new therapeutics. We will also be looking at recommendations and strategies to better get prevalence data, as well as collect surveillance and adverse event reporting. And very importantly, too, there will be also development and implementation of a regulatory framework should we define new food allergens in the future. And this will also include the scientific criteria. So you would think that now it's all settled, we have the allergens, but really that's not the case. Over 170 foods have been reported to cause IgE-mediated food allergies. And this is a list of some of the common foods that are on other countries' lists. Here, again, I have the list of the US list. Very recently, though, there has been some, and also has been highlighted that there needs, these lists have been around since the early 2000s, and whether we need to start updating or whether there are new allergens that may have a more global presence. And as of 2021, a recent report by the Codex, they formed an expert consultation, looked at the data again on potential allergens, and it basically confirmed most of the existing allergens, except for soy. Felt that the data for soy was not as robust as for the other allergens, but it did add sesame to this list. Also, it did define specific types of tree nuts: almond, walnut, pecan, hazelnut, cashews, and pistachios, and removed sulfites from the list because sulfites are not considered IgE-mediated food allergens. Again, though, there still are other issues. I mean, we're only talking about allergens in food labeled products. It does not involve drugs or cosmetics or other consumer products. And it does not, for the most part, involve foods that are sold in most retail or food service establishments, especially when you go order and they give it to you in a self-order type mode. And also another important area where the law does not address is with the unintentional introduction of allergens by way of cross contact. This has led to a lot of manufacturers voluntarily placing statements on products such as may contain, also called precautionary allergen labeling statements, PAL, to alert consumers of the possible presence of allergens. Now, this does not mean that these products are not regulated. If such statements are used, the FDA and other regulatory agencies would require that these statements are truthful, not misleading, and not used in lieu of good manufacturing practices. However, there are no authorities to determine when or what types of statements should be used. Nonetheless, because of the concerns of cross-contact, the FDA Food Safety Modernization Act was passed in 2011, and the final rule for preventive controls for human food in 2015 put allergen cross-contact on the map. It defined what that was, and that any firm using food allergens must establish and implement a food safety plan to control the risks and preventive controls for food allergens so that you can ensure accurate labeling of finished food products. Um, with this rule, though, there are no stipulations or thresholds established on when you would be able to say that your product does or does not contain allergen by cross contact. So there is a concern from the industry regarding, how clean the equipment and, how much allergen could be in the product. Um, and that would need to be declared. We have some guidance that's still pending on this argument, but basically these issues about labeling are a concern. Also, allergens and food avoidance risks. What we have learned is that from recent prevalence data that maybe up to 85 million Americans, or 1 in 4, are report that they avoid one of the top 9 allergens, with 1 in 10 at reporting allergy and putting them at risk for anaphylaxis. Food allergens appear to be everywhere, and there's seems to be a proliferation of PAL statements, but also we are seeing a lot more recalls, unfortunately, than previous. Allergens are probably now the #1 cause of food-related recalls that we are seeing. Another issue is that the food label allergen, you can see it there has the allergen, but that doesn't tell you what level of the allergen is in there necessarily, or amounts. There's different types of ingredients that contain allergens. But not only that, there are also multiple types of statements, PAL statements, that can tell consumers if allergen is present or not. And this can be a little bit confusing, but also more importantly, these statements are not typically linked to a risk. You can have a may contain statement that has a certain number of peanut presents, and the similar a facility statement with the same. It doesn't mean one is different from the other. And then importantly, I think as well, communication and perception of allergen risks varies. Most US guidelines do suggest that PAL be avoided by all individuals. However, surveys show that up to 50% are ignoring these statements. So which tells us that consumers are looking into these statements as possible reasons to consume products, or giving them some estimate of risk. 8 to 31% of accidental reactions are due to consuming PAL products, and most of these are recent studies, so showing us that there are concerns here. And while we have promising treatments, there is still no cure, so avoidance is still key. This paper, Defining a Path to Safety in Food Allergies, was by the National Academy of Sciences. Is a very, I would suggest reading this. This is very, highlights many of the gaps and difficulties of managing allergens, and it did identify that labeling could be improved, and one of these ways was to develop a risk-based labeling approach, possibly establishing thresholds. So before I talk about thresholds, I want to kind of define some key scientific terms. Hazard is what we typically call the intrinsic capacity to produce an adverse effect. So the hazard is the food, but the risk is how much of that food you're eating and to cause a reaction. So that's the likelihood of adverse outcome. And then when we talk about safety, we're usually talking about an acceptable risk. We can't always assume absolute certainty of no harm. So we have to have some acceptability. And an issue that has come about in the food allergen world is that we used to have a zero tolerance threshold. So if you assume that the shark, in this case, is the allergen, this is not a very real life presentation here, but this would be like a zero threshold where the shark is right at the edge of the water and is going to any allergen presence would cause a reaction. However, we are getting more data to suggest that this is more the likely picture, where I don't know if you can see it, but there is a little shark in there and it's close to water, but you might still be able to go into the water and not have a problem. And this is the challenge, is when do you get to these situations and how can you manage this? And this is where a lot of the data on thresholds at least is starting to inform us about these hazards and risks. So an allergen threshold has many different meanings, and it can mean different things to different people. There's the individual threshold. Many of you may have heard about this. This is your, the person with the allergy. That's their threshold. How much can they eat before they have a reaction, or how much can they tolerate, or maybe none at all. But when we're talking about more from a public health, we're looking more at population thresholds, which is basically looking at all the individual thresholds in the population to try to estimate some level of risk in that population. There's also an analytical threshold, which is just based on a test method. And then there's a regulatory threshold, which is basically putting on a limit or tolerance based on different laws or other public health measures. For allergens, where are the regulatory threshold considerations? Well, we have some possibilities to exempt ingredients from our labeling laws. Industry could come and show us data that their allergen or that their product does not contain allergenic protein, or is at a dose that does not cause an allergic response that poses risk to human health. So that kind of has a semblance of a threshold. And so data on that may help determine exemptions for ingredients. When we evaluate products for recall, they are given a level of class, recall classification based on class 1 if there is a reasonable probability of serious adverse health consequences, versus a class 2, which is a more remote probability of serious consequences, or unlikely class 3. So depending on thresholds and doses, we have started looking at different classes based on allergen presence. Evolving a little bit from the 0 threshold, like no, everything class 1, which most allergens are, by the way, class 1 hazards considered. But in some cases, when the levels are very low, the classification may be lower, and the question about need for recall as well. The advisory and PAL statements as well, as I mentioned to you, we have an issue of when do we need to put the statement on, and when would they be truthful or misleading? Is there a level? And then how clean do you have to keep equipment to keep that? And then, of course, if we do establish a threshold, it has to have good data quality, transparency, global consistency, and be practical and enforceable. The FDA has issued a document back in 2008 on approaches should a threshold. Now, the law does not require FDA to establish a threshold, but should we establish thresholds, we would be looking at different types of approaches. One is the analytical based, as we've discussed, which is based on a limit of detection. This has, to a certain degree, what we use for gluten-free products, which is set at a 20 ppm limit of detection. But we could, we are developing tests for different allergens, but I would say we are not there yet. We don't have tests for all the different allergens, for instance. But we are getting better suited to develop methods to detect a variety of different allergens in different situations to help us understand threshold responses. There's a safety assessment based approach, which is the traditional approach used in food safety based on lowest adverse effect level and safety factors. And then there's the risk assessment based approach, which is the most robust approach. And this is basically looking at risk in relation to dose, exposure, and possibly to severity. The good news is that in the food allergen world, as opposed to like microbial contaminants like salmonella or listeria, you don't feed people those foods and see what happens. But we do this all the time with food allergens. We do challenges in humans. So using that data, we can derive some dose response information that can help us in determining thresholds. And in this case, the threshold would lie between the dose and the escalation of the food challenge that caused a reaction, and then the dose that did not cause a reaction. That's where the threshold lies. And so it's in this interval. And using that information, when you collect enough data points from different people who have undergone challenges, you get this nice sigmoidal curve, of a dose distribution curve here. And what's interesting about this curve is that there is a huge variability within people in the population who respond to different doses. You can have people up here that are at like gram amounts versus people down here at microgram amounts, okay, millionfold difference. So you are able to identify different populations based on this curve, whereas the high dose responders are low sensitivity, low dose are high sensitivity. And very few, relatively few people are in this low dose. The problem is the diagnostics don't always get us there, and we need better diagnostics to help us determine who those low dose responders are. So we treat everyone on this curve the same. They have to all avoid allergens at all costs, and that creates a lot of issues in quality of life and so forth. Another important factor about this curve, though, is that you can start making some risk modeling determinations based on percentage of people at risk. So here you can see this is the ED20, so that's 20% of population is going to react at this dose, ED5, 5%, and so forth. So this helps risk assessors understand possible dosing problems to allergens. And indeed, this is taken from the VITAL 3.0. This is the Allergen Bureau of Australia developed a risk modeling approach for all different allergens. And what these numbers are based on are the ED1, or 1% of the population to react. You can see that for the most part, the numbers are pretty consistent for most of the allergens. There are some outliers here in terms of numbers, but for the most part, this is down at the low levels. And you can see also here that some data is better than others. You've got more data points for some foods like egg, milk, and peanut than for other foods like mustard and so forth. But I'm just showing you this, that there are methods to try to estimate dose thresholds. I know many of you might not know what this means, so I always like to show a little bit of a visual. I like this graph here of just showing you what an ED1 would look like. So it is a little bit like a pebble, a few pebbles of sand in there. It's a very low amount. But what's important too when you look at these risk modeling studies is that when you get up to a 1 peanut, which is about 400 milligrams, that's still 40% of the population. So probably half of the population can consume a peanut or more and not have a reaction. And so we need to do better jobs of trying to understand who those people are versus, obviously people down here reacting to these doses are in a much different risk category than people up in this category. Yeah, so there are some threshold data limitations that need to be considered. As I mentioned already, there are some better data, more data endpoints for some foods versus others, but there are also many of these data sets are from different populations, some geographically different, but also using different types of methodology to understand dose responses as well as stopping criteria and so forth. And when you're looking at some of these studies in particular, there can be differences in threshold determinations for different foods depending on which criteria used. One example is a recent study that was published sponsored by ILSI by Haber et al., which showed a lower threshold for peanut compared to other estimations. There's also issues about reproducibility of thresholds, but also importantly, though, is that this information doesn't provide severity information. It just tells you what is a percentage of people reacting to that dose. It could still be a severe dose, and is that really a risk to human health or not? And so we need more information. Here, the study, the PATS study, showed basically they did a proof of concept. They took the ED5, which is the 5% dose, I think it's 1.5 milligrams of peanut protein, and gave it to 350 peanut allergic consumers from 3 different geographic areas. And they found that actually they couldn't even get up to 5% of people to have objective reactions to this dose. It was more like 2%. And none of these individuals actually had severe reactions. Uh, Other types of data looking at peanut in particular has not identified severe reactions less than 2.5 or 1.5 milligrams, or at least life-threatening anaphylaxis at these doses. The recent study by Turner et al. Also showed a similar pattern for milk. So there is maybe some information that maybe these lower doses are not as severe. Nonetheless, though, most people in the population just don't know what their thresholds are. Here, we sent a request for comments and information on risk assessment back in 2012. We had didn't get so many submissions, but most of those submissions were from concerned consumers who did not want the FDA to establish any thresholds. Most industry and trade groups were in support, but not necessarily the consumer groups. Um, Another survey by Marcus Soto, it all showed that people, less than 40% of people, when asked, did not know what a threshold was. So very important is that, though, has been a food allergen and risk assessment consultation by the FAO and WHO, which is looking at addressing thresholds, and they have met as early as March of this year to talk about thresholds, and I think we're going to be seeing some interesting data coming out from that soon. So just to end my discussion, what are some of our future focus? Well, I think the implementation of the FASTER Act is going to be a very important process. Not only will we be looking at adding sesame to our list of allergens, but also exploring some science around food allergies to improve labeling and empower information on allergens. We are looking at recalls and what can best to prevent recalls with our preventive controls and through FSMA, and then foster global discussions on thresholds, risk-based strategies to address cross-contact. And I think a very important part is going to be educating the consumers about this, and some more information about our food allergy efforts are at this link. And thank you so much for your attention.
Hello, I'm Bruce Roberts, Food Allergy Research and Education's Research Strategy and Innovation Officer, and it's my pleasure today to tell you about recent developments within the FAIR Clinical Trial Network and our activities to advance treatments and preventative therapies for food allergy. My disclosure is shown on this slide. For the purposes of today's presentation, it's really two parts. In the first part, I'll be describing recent capabilities we've established within the FAIR Clinical Trial Network that relate to our patient registry, the FAIR Clinical Network itself, the biobank that we have established, and a data coordination center. And then I'll be moving on to describe specific research initiatives that we've launched which leverage these capabilities which we've established. I'll move on to our patient advocacy activities and describe most recent activities related to the FASTER Act. And this will set the stage for the final topic, which is really leveraging everything that I've described in our capabilities towards a new initiative to evaluate information, oral food challenge data, to assist in efforts to establish allergen threshold levels. So a few words about FAIR. FAIR is the world's largest private funder of food allergy research, driving innovation towards making effective patient-centered approaches for prevention, diagnosis, and treatment of food allergy. We're modernizing food allergy diagnostics in an era of molecular diagnostics and medicine. And we're providing pediatricians with the tools to educate parents about introducing potentially allergenic foods to infants at risk of developing food allergies. Now, what I'll be describing in the first part of the talk is these core competencies that we've established, which are shown here in this foundational graph, which is the patient registry, the clinical trial network, and the biobank. So let's turn to the FAIR patient registry first of all. It's the largest US food allergy registry with some 12,600 children and adult participants, and it's growing. It brings the patient voice to food allergy researchers, accelerates the search for new treatments, and provides life-changing improvements for patient care. Through online surveys, registry participants can provide vital information that help us to better understand food allergies and the patient journey. And we're currently focusing attention on patient sampling to ensure that we maximize representation of minority groups within this registry. Now, a few facts just with respect to the registry itself. So the relative breakdown of adult to children is about 43% to 57%, and the age range spans everywhere between less than 1 year of age up to more than 80 years of age. 45% of the respondents are male, 55% female. And using the information that have been provided by this information, we can query it in order to get some key information with respect to food allergy, the frequency of it within the North American population, but also other pieces of information which are really critical to understand, such as 79% of the individuals were diagnosed with food allergy in childhood. We track over 200 allergens in total in our registry. Now, importantly, 78% of the individuals are allergic to more than one food. And this really, I think, exemplifies the fact that we need to be thinking about therapies that potentially can be used to address the totality of the disease. 45% of the individuals have one or more reactions on an annual basis. And interestingly, 48% suffered their last reaction at home. So I think this also tees up a really critical point in this space. With individuals who are diagnosed with food allergies, in many circumstances, because we only have one approved therapeutic agent, then the guidance is avoidance. Now, even when individuals make an absolute effort to try and avoid particular allergens, it may be that they're still going to suffer adverse events because of accidental exposure. And so this is, I think, an issue that we are aware of, is that avoidance isn't entirely a solution because it doesn't entirely protect individuals from encounters to allergens. So let's turn to the clinical trial network. So this is a consortium. We're bringing together top institutions to ensure that patients have access to state-of-the-art care and diagnosis. And we're committed to the following: the development and dissemination of best practices for the care of patients with food allergies. The sites serve as places for the execution of clinical trials for the development of new therapies and for diagnostics. We're engaging in using cutting-edge technology and research to better understand the biology of food allergy. And this provides a rich source of information to help us with the patient registry and the biobank. So a bit more detail is actually provided on this slide. So the FAIR Clinical Trial Network, which has now been newly expanded, consists of some 50 sites all throughout the United States, and their geographical location is indicated on this map. And importantly, collectively, these sites see 115,000 food allergy patients on an annual basis. So you can imagine that this is a very rich source of information that we can leverage to understand issues that are facing patients, access to care, but also to help us with clinical studies, whether they're observational studies, prevention studies, or intervention studies. Now, turning to the biobank, our biobank, which we've established in conjunction with Cincinnati Children's Hospital, provides a range of biobanking and biomarker services to support multi-center investigations. And many of the services that they provide are actually shown here on this slide. Now, by virtue of the fact that we have these three capabilities, the patient registry, the clinical network, and the biobank, it affords FAIR the opportunity to catalyze the advancement of clinical studies. And in that regard, we really think of ourselves as being able to serve as a catalyst by providing services in the area of planning, execution, and evaluation of clinical studies. And that's shown in this slide. So we built this capability not only to support the advancement of new therapeutics that may be offered by pharma and biotech, but also to support ourselves in our efforts to basically champion some new initiatives, which I'll be describing presently. Now, to pull all this information together, you need a data coordination center. Recently, we've established such a center in conjunction with Dr. Jonathan Spergel at CHOP, and they support critical FCN activities with the design, development, execution, and monitoring and analysis of clinical research. And many of the core capabilities are shown on this slide. So now I'm going to turn to the aspect of the research initiatives. And the key thing to think about in this regard is we believe that FAIR can serve as a catalyst for the advancement of particular initiatives that would otherwise not receive much support or attention were it not for FAIR's participation and drive. And this slide shows how that base capability, the registry, the network, and the biobank support these 4 priority initiatives. And they are as follows. In prevention, we're very keen to pursue new therapies that might block the development of food allergies in the first instance. We're pursuing new therapeutic applications which may lead to long-term desensitization of individuals to multiple allergens. We're focusing very heavily in the area of understanding disease biology, a particular investment in the FACI-Broad initiative to understand the role of the gut-brain axis as it relates to food allergy. But the one thing that I wanted to highlight today for the purpose of today's presentation is diagnostics and assays. And in this regard, FAIR has recently announced what we call the Faith Diagnostic Challenge. And what it is a competition designed to basically encourage investigators to come up with alternatives to the oral food challenge. It's a multi-million-dollar competition designed to promote and develop a diagnostic test or tests for food allergy. It's intended to promote innovation and encourage consortia of investigators to work together to devise new solutions. And more details related to the Faith Diagnostic Challenge can be shown at the link below. Now, turning to patient advocacy, FAIR has historically played a major role in terms of patient advocacy and the generation of new policies and legislation that protect the rights of food allergy patients. And in this regard, FAIR played a pivotal role in terms of the passage of the FASTER Act. This was accomplished on April 23rd of this year. It's a new law that requires sesame to be labeled on all food packaging by January of 2023. And the law also requires the government to consider a research report on food allergies and establish a risk-based scientific framework for adding future allergens. FAIR played a multi-pronged approach in terms of getting this act passed, which is actually indicated in here, from participating in congressional hearings to then working with lawmakers to actually see that this actually was passed into law. And the impact here is that we were able to help the nearly 1.6 million individuals who are allergic to sesame in a period of just less than 2 years that led to the passage of the law. Now, coming back to this aspect of a scientific framework for adding future allergens, the key aspect of this is thinking about allergen thresholds, which tees up the last piece and the topic that I wanted to discuss today, which is our new initiative in this space. And the reason why, in fact, I went into the detail with respect to the existing capabilities is that we're going to harness and leverage all those capabilities to help us in this regard. So let's stop and think for a minute about what we're talking about here. So what constitutes a safe level of allergen within a food? That's the fundamental question. Now, in the absence of guidance, we use precautionary labeling, but that's not particularly informative or helpful for consumers or patients. What has been done is clinical data based on the assessment of food allergy patient allergen sensitivity has been employed to establish so-called preference doses. And the way that that has come about is leveraging oral food challenge data, which has become the gold standard for assessing the sensitivity of individuals to food allergens. But there's an issue in the United States. There's a paucity of standardized oral food challenge data which has been generated at US clinical sites for the top 9 allergens. So this is where I think the FAIR can actually serve a role in terms of garnering and collecting that oral food challenge data from multiple sites within the US. The oral food challenge involves oral administration of a known quantity of a potentially allergenic food on a blinded basis to a subject, followed by documentation of clinical responses noted within a defined time period. Now, depending on the nature and the severity of the response, the subject can receive successively larger portions of the food until a response or no response is noted. And responses can encompass a myriad of different symptoms, including skin, respiratory, gastrointestinal, cardiovascular, neurological, oral, and ocular signs. Now, while there are some 32 million food allergy subjects within the United States, oral food challenge data is only available for a subset of these individuals, and there's several reasons for this. First of all, subjects can be diagnosed as having a food allergy based on a combination of other factors, such as their clinical history, skin prick, and IgE test results. And so no oral food challenge may be available for that individual. Another aspect is that the oral food challenge needs to be monitored by a clinician because there's always the potential for an anaphylactic reaction, which is why some individuals have not undergone an oral food challenge. And finally, the test itself can be time consuming. So for this reason, our expectation should be that only a fraction of food allergy patients have actually undergone an oral food challenge test, and therefore the results might be available to potentially use and help with allergen threshold calculations. Now, there's two aspects that we need to consider in this regard, which is the presentation of the allergen and the response of the individual. So see, these are some of the things that we need to think about because this isn't a straightforward exercise. First of all, for the given food, there's the quantification of the amount of allergenic protein. And that's going to be dependent on the sampling method, the consistency, and the experimental error. Then there's the means of reporting. Protein concentration, was it described or was it described in terms of the size of the serving? How do you equate that to the concentration of protein that was actually delivered? The form of the protein preparation can also have a bearing, as can the source of the protein itself. And then there's the definition of the units. Is it by parts per million, by weight per weight of serving? So when we actually get oral food challenge data, we may actually see the different sites report the findings in a number of different ways. And they may actually record the response of the individuals in different ways, or that in fact the results may actually need to be considered in light of a number of considerations. So oral food challenge is the gold standard methodology for testing for sensitivity, but it's done in different ways at different sites. And so standardization of oral food challenge testing is really important here. Then there's the question of what constitutes a clinical reaction. And as I mentioned previously, this is complex. It does require a trained observer to actually determine whether something really qualifies as a clinical reaction. There's the variability of the response within a given subject. So depending on their physiological condition, you can get different levels of response depending on whether they're stressed. Then you get variability response within the patient population itself. Different individuals have different sensitivities to particular allergens. And so you really need to then understand, so what is the response in a generalized population? Which then tees up the question is what's the sample size required for each individual allergen? And finally, there's the whole process of taking the oral food challenge data and then translating that into threshold levels and the criteria for doing that, which is beyond the subject of today's presentation. Now, just going back to this point, I just wanted to cite recent work by Dr. Ruchi Gupta, who's reported that the prevalence of food allergies within children can vary significantly. So if you're talking about peanut, milk, shellfish, it's likely that there will be a lot of oral food challenge data available for subjects because these are the more prevalent food allergies. But if you start to talk about less prevalent food allergies like wheat, soy, and sesame, there may be fewer oral food challenge data points upon which to then gather information to establish allergen threshold levels. So this needs to be borne in mind in an exercise of this sort. Now, fortunately, the VITAL-2 group has actually reported benchmark results that help us give some insights in terms of what needs to be done here. This is data that was largely gathered at clinical sites outside of the United States, sites in New Zealand, Australia, and in Europe. And they gathered information, reported allergen threshold levels in 2011, and then subsequently refreshed that data in 2019, essentially doubling the number of observations per allergen. There's a couple of key points that came out of that report, is that the threshold value for peanut really was unchanged between 2011 and 2019, which was interesting, in spite of doubling the total number of observations. Interestingly, threshold levels for milk, sesame, soy, and wheat didn't change by very much, even though the number of observations had doubled. There was a more significant change in the case of egg and shrimp. But one take-home message from this is that as few as 50 oral food challenge observations may be sufficient to establish a threshold level for a given food allergen. But obviously, more data is actually going to be more powerful in this regard. One thing that we really need to think about in terms of the food allergy research community is that data likely to be reflective of the entire food allergy patient population? So the more observations we have from more subjects, the more confidence we're going to have in our threshold values. So this is where I think FAIR comes in. And what we're doing now is launching an initiative in order to pull together information from clinical sites within the United States. And there's two parts to this: the collection of historical data, So we're collecting oral food challenge data from the clinical sites within our FAIR clinical trial network of 50 sites in conjunction with the FAIR Data Coordination Center at CHOP. De-identified patient data will be collated and standardized to allow for pooling of the observations. So this is looking back historically. This is not going to be an easy task because, as I mentioned at the outset, individual clinical sites can record their data in slightly different fashions. And in some cases, information related to the quantity of the allergen actually delivered to a subject may be missing or lacking or not presented in a standardized form. So we basically will need to do some scrubbing of the data in order to enable us to basically do pooling and a determination of an ability to actually compare results from site to site. The second part of it is the prospective component. So we're developing standardized methods for the performance of the oral food challenge and recording of data in conjunction, again, with the FAIR Data Coordination Center at CHOP. And this will lead to standardized methods for the dissemination to all the FCN sites to facilitate oral food challenge data in future clinical studies. So this is not an initiative which will be short-term. This is much more of a long-term initiative in which we'll be working in conjunction with our sites to create a database of oral food challenge data from all of the sites within the United States that are participating in our FAIR clinical trial network. And an important aspect of this is we anticipate that we will probably realize there will be gaps in the data, and that may then necessitate the requirement to do particularly targeted or focused clinical studies to specifically gather more oral food challenge data in instances where there's a paucity of observations for a given allergen. So that concludes my presentation for today, and I'd be very pleased to address any questions that you may have on any of the FAIR initiatives. Thank you very much for your time.
Hello, everyone. I'd like to thank the meeting organizers for the opportunity to speak with you today on opening the allergy black box. It's an honor to speak with you and with the fellow panelists. So first, I'd like to start with my disclosures, and while you're looking at those, give some personal background. I'm trained as a data scientist and mathematician, and developed many commercial medical tests in the areas of oncology, pregnancy, and food allergy. And the work presented here today is a collaboration between Allergenis and Hugh Sampson at Mount Sinai. For Allergenis, the mission is to use innovative technologies for monitoring immunity, partnered with machine learning techniques to bring more confidence to the diagnosis and management of food allergy. Let's preview what I will be talking to you about today. So first, challenges in food allergy, and then moving more specifically into assessing peanut allergy sensitivity. And then looking at some recent technological innovations and the resulting opportunities that these give us, in particular as they apply to the development of a risk-free sensitivity test. And then finally, closing with some of the implications of this sensitivity test for various stakeholders. So many of us are aware of the challenges that face us in food allergy, from all of the cases of kids moving or going to the ER, the prevalence of children with multiple food allergies, the long-term risk and immediate risk of having a food allergy and comorbid conditions like asthma and eczema. And then, of course, the economic cost of food allergy, which is in the billions of dollars. But beyond this, I'd like us to focus today on the family and the everyday struggles that families face. So things like just buying groceries, daily meal preparation, life outside the safety of the home, access to healthcare, and coordinating events and coordinating schools, among many, many other things. And often what goes overlooked are the secondary implications. So the impact on caregivers, on parents, So the mental and emotional impact of food allergies on parents, in fact, is frequently greater than the impact on their children or on patients. Food allergies have considerable impact also on the social lives of parents and can cause significant financial strain and, of course, time burden on parents. So food allergy and impacts the family significantly in a variety of ways. So in food allergy, there are some very basic questions that remain unanswered. The first is diagnosis. Does my child have a food allergy? And as many as up to 60% of food allergy sufferers may be misdiagnosed, which is enormous when you think about the strain on these families when, in fact, the children don't even have a food allergy. But in those cases where there is a food allergy, there's the sensitivity question. That is, if my child is food allergic, how allergic are they? And how does this sensitivity, the degree of food allergy, impact their day-to-day lives? Is my child hypersensitive? Could they have a severe reaction even to a minute amount of peanut? Or are they less sensitive and because of this, maybe less anxiety or less vigilance around the food allergy? So let's take a look at the sensitivity challenge. So in theory, sensitivity is determined by the oral food challenge, or OFC. So in the OFC, a child ingests increasing amounts of food allergy until a reaction occurs. That's called the reactive dose. The dosing regimen typically follows a guideline like Practol, where first they start at a low amount, 1 milligram, then they move to 3, graduate to 10, 30, and so on. And at some point, let's say 100 milligrams, they have a reaction, and that's their reactive dose. And that's considered to be the amount of food allergen that the child can ingest before they have a reaction. And that's their sensitivity. So highly sensitive children react at 1 or 3 milligrams, and less sensitive children may react at 1,000 milligrams. But in practice, the oral food challenge is rarely used to determine a child's sensitivity level for many reasons. One, it's dangerous. We're having children ingest the food that they've been told to avoid. The oral food challenge is unavailable. So typically they're performed at only at certain centers where they can deal with the negative side effects, reactions that can occur because of a child ingesting, let's say, peanut. Or mainly because of unwilling parents or physicians or children, just due to the risk of performing an oral food challenge. So all of this results in what I call the black box. So for the question of how allergic is my child, most often we just don't know. And in the absence of this knowledge, avoidance is the only safe course of action. So we don't know how allergic your child is, so don't ingest any peanut at all. So in my personal experience developing tests in oncology and pregnancy, this lack of insight into a fundamental question around the condition would be shocking. And that's where we stand in food allergy. One is the diagnosis question, which we have made some progress on, but we still need to be able to answer that question with more precision. But we don't have an answer to how sensitive is a child. And it's even more complicated than that. So on the left, we have this question that we've been talking about, how allergic is a child? But on the right, there's the food that the child is ingesting, and the question is, how much allergen is there in the food that's on the shelf when the mother or father goes shopping? So this brings in 3 parallel relationships one between the child and the food, how allergic is a child and how much allergen is there in the food, but also between the parent of the child and the food companies producing the food, and also between those allergy researchers trying to understand how allergic children are to those food industry researchers trying to understand how much allergen there is in the food that's produced by their companies. So really we have a two-body problem here. So today we're going to focus on, I'm going to focus on how allergic is a child and try to answer that question and talk about the progress that's been made on answering that question. I suspect others at this conference will be speaking about how much allergen is there in the food. So one of the questions, the main question that Allergenesis is looking at is, what if food challenges could be conducted ex vivo? So the oral food challenge is in vivo. We actually expose the child to the allergen, in which case there are clinical reactions, and that's actually part of the challenge is we feed them more and more increasing amounts of allergen until they exhibit some reaction. What if we could move away from that approach to a blood-based test where blood is taken from the child, there's no exposure to the allergen, and we assess their sensitivity from their blood as opposed to having to actually expose them to the allergen. So Allergenesis' objective is to provide an ex vivo alternative to the oral food challenge to enable frequent, safe, and accurate diagnosis and assessment of food allergy sensitivity. So imagine not just, in fact, a single sensitivity test, but being able to monitor sensitivity over time, let's say every 3 months, and determine improvement in a child's sensitivity due to diet, outgrowing the allergy perhaps, exercise, or desensitization through therapy. Right now, there's not a tool to measure any of those, the impact of any of those. I'm not going to go into the science, just one slide on the technology behind this. So the way the Allergenesis technology works is through a innovative technology called epitope mapping, which has been developed at Mount Sinai. So basically, there are 3 steps here. The first on the left is to take a patient sample, which is really a sampling of the immune system containing the repertoire of antibodies that a patient has, a child has, and also the allergens, in this case from a peanut. We know the proteins within a peanut that the immune system of a child reacts to. And on those proteins are regions called epitopes. And it's these epitopes which we've identified, which allow us to diagnose which children are allergic and, in fact, how allergic are they. This is very much has an analogy to COVID, which most of you, of course, are familiar with. With the spike protein, also has epitopes which react with the immune system. And it's in fact that interaction between those epitopes and the immunity that's built up through, let's say, vaccination that allows that reaction, immune system reaction to not occur. So very much the same here. And what we've done is we've taken this in vivo reaction within a child, and we've moved it outside by taking a sampling of the immune system and taking these epitopes, creating them artificially, and putting them to an instrument, a Luminex instrument, and allowing those in a test tube to come together and measure that reaction. And it's measuring this reaction that, on the Luminex system that provides the data that we can then apply machine learning algorithms to answer questions like, is this patient allergic or not? Do they have these epitopes that react to the immune system? And if so, how allergic is the patient? So the allergen sensitivity test determines how allergic a patient is. So in fact, this approach, epitope mapping, has already been addressed for the diagnosis question, and a paper referenced here on the left in collaboration between Allergenis and Mount Sinai primarily has just recently been published this past month, showing a 92% sensitivity and 94% specificity, which is the most significant advance in peanut allergy diagnosis in 10 years. So coming back to this 60% over diagnosis of peanut allergy, it reduces that by tenfold. So this alone provides parents with some hope that many of these children that are diagnosed with peanut allergy, in fact, are not allergic to peanut, and hopefully alleviate some of the anxiety around peanut allergy. However, that still leaves many children who truly are allergic to peanut. And for those, the question is, how allergic are they? And for the rest of this talk, I'd like to discuss the sensitivity test and its development. So the development of this test has many elements. I'll just touch upon some of those. This, by the way, is not published yet, and it's work that's just been completed and will be published soon. So first is rigor. So we follow the National Academy of Medicine guidelines for test development, which really focuses on independent discovery and validation sets, blinding, and using proper stats to come to conclusions. Well-characterized samples following guidelines like the Practol standards, double-blind oral food challenges, so very well-characterized samples in terms of sensitivity. Generalizability, so multiple sample cohorts, 4 were used here. Often we see 1, maybe 2 cohorts used in the development of a test. Here, 4 cohorts from around the world, including 2 here in the US. So diverse demographics, geographies, and ages in the development of this test. Quality, extremely important. So all of these, the development of this test done in a CLIA and CLSI laboratory, guiding best practices, and appropriate documentation for clinical practice grade test. So none of this work has been done in academic labs. It's all been done in a regulated laboratory. And then finally, of course, the innovation that Allergenis is bringing to the table, and that is epitope mapping combined with advanced machine learning for the next wave of food allergy tools. So the approach we take is that, again, advocated by the National Academy of Medicine, and that is starting with patients with a range of food sensitivity levels. So those who are very sensitive, 1 milligram, let's say, to those that are not that sensitive, 1,000 milligrams. And then apply machine learning algorithms to predict the sensitivity level from the epitope reactivity level. And then take that learned machine learning algorithm and then apply it to a totally independent set of patients and assess the performance. In developing this test, what we learned is that it's ideal to first put each patient into a sensitivity level from very high, that's row number 1 here, where the reactivity level is below 100 milligrams. So maybe reacting at 1 or 3 or 10 milligrams. to high, 100 milligrams to 300 milligrams, keeping in mind that a peanut is around 250 milligrams. To moderate, 300 milligrams to 1,000, and then low sensitivity being over 1,000 milligrams, so over, say 4 peanuts. So the machine learning algorithm predicts the sensitivity level for a subject first. So are they very high, moderate, or low? And then characterizes each of these sensitivity levels, which I'll show you in the next few slides. Important to know, this validated test has a 90% accuracy for predicting which sensitivity level a patient falls into. Keep in mind, to date, there has been no test or tool beyond oral food challenge, which is avoided, to be able to ascertain, is my child in the very high group, very, very sensitive, or in the low group, much less sensitive. So characterizing each of these 4 levels. So in red here on the left, we have level 1 or bin 1, those children who are very sensitive. To blue, which is level 2 or bin 2. In black, level 3, and in green, level 4, the least sensitive. These are log normal distributions, and they're based on the predicted level of from the population in each of those groups. So the mean, standard deviation, and other characteristics of that population. It enables the estimation of the probability of an allergic reaction at various allergen levels. So, for example, if we take a patient in the green in bin 4 who's not very sensitive, and you compare their reaction profile to those in red in bin 1 who are very sensitive, there's very different information here. So, for example, a child in bin 4 may have a reaction at 750 milligrams, which is about 3 peanuts, whereas those in red may have a reaction at only 30 milligrams, which is about 1/8 of a peanut. So you can imagine being a parent or a physician and wanting to know very much, is my child in the red bin and very sensitive, and that's going to affect my decisions in preparing meals, buying food, and so on and so forth? Or are they in the green bin where they're really actually not that sensitive to trace amounts of peanut, and so my buying habits and meal prep habits would be affected, my day-to-day habits would be affected knowing that. So going into these bin characteristics even more, so for parents and physicians, they're particularly interested in knowing what is the smallest amount of allergen that may cause a reaction, and also conversely, what is a safe amount of allergen. So if we were to look at these 4 bins or 4 levels again, the median reaction amount, 46 milligrams for level 1 up to 750 milligrams, or about 3 peanuts for level 4. But importantly, what is the smallest amount of allergen, in this case peanut allergen, that a child would react to using the 95th percentile. So for level 1, it's just 3 milligrams, so very sensitive. Whereas for level 4, it goes up to 200 milligrams. So there's quite a dynamic range there. Again, a lot of information from being in level 1 to level 2 to level 3 to level 4. Another way of looking at this is, well, what's the probability that there would be a reaction at 1 milligram of ingestion? Well, for level 3 and 4, the risk is basically 0%. In level 1, it's 0.7%, and then level 2, 0.3. So still very low, but not 0. So this allows us to quantify degrees of risk for a patient, for a child, whereas again, prior to this, this information was just unknown. And so finally, I'd like to close with what some of the implications are for various stakeholders. First, for parents and children, there's more information than simply, sorry, you just need to avoid peanut. There's the impact on food for shopping, meal planning, and just the perception of risk within the household and outside the household. And thirdly, sensitivity can be now known without an oral food challenge, simply with a blood test, and it can be monitored over time to see if there's change. For providers, recommendations on avoidance or not could change. Management of treatment options, so oral immunotherapy options are now available, which children are candidates for immunotherapy, which would benefit, which ones would very much benefit from going on treatment based on their risk profile. And then finally, recommendations on safety food challenges. So often knowing if a child is in, say, bin 3 or 4, that is their sensitivity is not that high, a safety food challenge may be provided at a safe level to see truly how safe that child is. Would they react at 3 milligrams if this sensitivity test show that they should not react at that level? And so that would give the parent and the physician and the child even more confidence to go forward, knowing that they could ingest safely a certain amount of food allergen. And of course, for the food industry, just to be aware that technologies are now coming online that enable a child's sensitivity and a parent's shopping choices to be more informed, and how to take this information and utilize it in the efforts in the food industry to label food appropriately. What are the kind of labels that would be relevant for a parent who has this information about their child and sensitivity levels? How could that be helpful for the consumer? And so with that, I'd like to acknowledge the teams at Allergenis, Data Insights, and Mount Sinai, and just thank you all for your attention.
Well, great. Thank you all for the interesting talks. And I'm going to ask Dr. Roberts and Dr. Kearney to turn on their webcams. There's Dr. Roberts. And there's Dr. Kearney. Hello, great. So to the audience, so unfortunately, Dr. Lucioli was unable to participate in the Q&A. And I did note some questions come in that were during his talk and maybe specific to his talk. And we'll I'm sure the IFEN staff will capture those. But for the sake of kicking it off, I thought one of the questions was interesting, and I think I'd be interested to hear from both perspectives here as it relates to the sort of the new, I'll call them the new recommendations, within dietary guidelines on introducing the early introduction of certain allergenic foods. So for example, peanuts and eggs. During the first year, and that's that's a shift in some of the guidance that had been given historically, and I'm wondering from a from a consumer perspective how that resonates and what that looks like within the affected populations. So I don't know if Bruce, if you want to kick off on that one.
Thank you. Actually, thank you very much for the question. So just to sort of take a step back here, there are really two key studies in this area. The LEAP study was a very successful study conducted by investigators in the UK who demonstrated that early dietary intervention with peanut could be successful in reducing the frequency of food allergy. The subsequent study was a more ambitious study, EAT study, which involved 6 allergens and was seeking to demonstrate the same point. The challenge is that as you increase the number of allergens, just from a compliance point of view, it can be challenging. And so really there has to be a commitment on the part of parents in order to stick to the regime. And in some cases, you're talking about feeding 2-3 times per week, with a portion of that potentially allergenic food in order to see benefit. So this, I think, is a very promising area, one in which I think the observations with peanut are really unquestionable. They achieve their primary endpoint. And now we need to see whether, in fact, that approach can actually be applied to other allergens as well. In principle, it should be the case. But there is an aspect of the window of opportunity, and I think to your point, Jason, is that we think that you really need to get in as quickly as possible in order to be able to condition the immune system to be tolerant against these particular food allergens, and that's the key. But there's another aspect to this, which is just in terms of the logistics, and that in enabling parents to basically stick with a friendly approach in terms of try to introduce the allergens over a period of time in order to see a successful outcome here. So in that regard, it's one thing that FARE is actually very committed to do, is to support in terms of getting more understanding in this area. But it is interesting that the guidance with respect to peanut is already out there, and it is somewhat surprising how parents are really not abiding, by that guidance, is that they're not really bought into the fact that early dietary intervention with peanut is potentially going to be successful. So there's also an education component to this that we need to understand, is demonstrating the point with clinical data, but also ensuring that people have access to the educational materials so they appreciate the potential upside and benefit of actually early dietary intervention.
I'll just add, agree with everything Bruce just said. I think I covered it well. I'll just add that I think it also underscores what we've, in all 3 talks we heard today, is that when we introduce therapy, either through regulated therapy or maybe a less formal therapy used at home through the early introduction of say peanut or other allergens, we need technologies or tests to be able to measure the effect, and that will give confidence over time to families that yes, we can make a difference. So without tests or technologies to measure, for example, the reactivity level of a child before and after formal therapy or informal therapy, they still don't know if there's been a benefit or not. So I think it underscores all the aspects of the 3 presentations today, the importance of understanding and be able to quantify where a child or an individual sits in terms of their reactivity level.
Yeah, I mean, I have to think that just the fact that all the various stakeholders within the allergic community are, continue to discuss thresholds. I mean,
Yes.
that's got to be taken as a positive sign. I mean, it's been a conversation for many years. I do start to wonder if thresholds, if there's a better term, because it can, I think Dr. Lucioli identified, it means different things to different parties, right? And maybe from a consumer's perspective too, I think that arming consumers with an indication, and Paul, with what you presented, simplifying it really into various bins or segments. I mean, that's one thing, but will that instill that level of confidence in the consumer? Like, well, at what point does the consumer proceed with some degree of certainty?
That's, I think, a fantastic question, and I think it's really important. And I know we have done a lot of work of talking to consumers talking to all the parent groups out there, and what do they want? Not what do the clinicians want or the researchers want, but what do the parents want? And if we can connect what we're all doing from a research, a strategic perspective to what the day-to-day that parents are facing when they go into the grocery store and they're trying to decide, what can they buy or what can they not buy? Are they adhering to strict adherence avoidance, whether that's needed or not? And just managing the day-to-day. When we can tie what we're doing to their day-to-day, that's when they start paying attention. They start figuring out how they can tie, whether it's a test or a strategy or a therapy, to their day-to-day, and how will it affect, and this is really clinical utility, is a term that we use, but how will it affect their day-to-day? How will it make their lives better? How will it reduce anxiety for the family in managing food allergy in the home? That's when you get buy-in. That's when people pay attention and saying, okay, this will affect my life. This will improve the life of my child. This will improve the life of the family. So I think that's key for adoption.
Okay. And for the folks on the phone, I'm going to take the liberty here. I think our speakers have agreed for a few minutes of overage. So I just want to hit a couple more questions. So if you can stay on for a couple more minutes, that'd be great. One of the questions that came in, it relates to FAIR's threshold, I guess, database to build. And Bruce, is there a sort of target number of participants like that you're aiming for in that regard?
Well, I think it's an important aspect that we need to look at is, and this is why I made reference to the VITAL data, is that they looked at the information and they parsed it by age and by geography. And they really didn't see that it made a fundamental difference in terms of the allergen threshold level that they determined. But they haven't really done it based on other demographic factors. And so coming back to Paul's point, I mean, we recognize that there's different sensitivities, and there may be different sensitivities within different patient populations. So if you look at the VITAL data on face value, it may say as few as 50 observations may be sufficient to give you a valid allergen threshold level. And then if you double that, it doesn't really fundamentally change. What if you change the composition of the patient population that you're dealing with? That would change it. So I think that we have to be mindful of the fact that we would probably want several hundred data points, from all across the United States, and try to make sure that that data is actually coming from a patient population that's representative of the diversity within the United States, so that when we look at that, we could actually say, is there any differences between different backgrounds? And also, other conditions as well that might be contributing to the effect, so that the value that we come up with at the end of the day is ideally a universal number. It's really reflective of the whole patient population. So we're really not, I think, in that situation in terms of looking at the data points that we have right now, because even with Peanut, where you have over 1,000 observations, you have to ask yourself, do we have the demographic information? Do we really know that's representative of a larger patient population? We don't know that. So coming back to the FAIR Clinical Trial Network, we believe that we're in a position to actually get thousands of data points through actually accessing our clinical trial network. And that would really give us high confidence. But it'll be a stage-wise process, just like VITAL. So if we have 100 observations and we go to 200 and the number doesn't fundamentally change, and the population is essentially representative, hasn't changed between the 100 and 200 level, we may be comfortable at that point and then come to the conclusion that it's not really necessary to gather other data points. But I think the big fundamental question in this area is your data representative of your patient population?
Yeah, well, and I guess that kind of lead for both of you on this kind of a second part of that question was around the need to sort of revisit the threshold data, and maybe at an individual level. And is that something that would be necessary? Maybe like looking at or would you say from your static demonstration of demographics, you'd have enough to go on? And I think, Paul, from your perspective, is that type of consideration built into, say, the machine learning aspect of that kind of modeling?
It's a really good question. When we look at the world from the oral food challenge perspective, there's so many other variables that come into play due to the nature of the oral food challenge. It's done in the office, anxiety's high, especially if there are children involved. They're being asked to ingest something that they've been told all their lives is a poison for them. When we use the oral food challenge as the gold standard, which makes sense right now, when we measure the reactions, we're not just measuring the biological reaction, we're measuring the anxiety and all these other effects. And some recent publications have come out that have shown that those other effects are actually have a pretty big impact on the output of the oral food challenge. People may react much sooner or earlier in the oral food challenge than at a lower dose than what would be expected. That said, if you move to, say, as we're doing at Allergenis, to a blood test where we're trying to eliminate these other factors and measure, what's the sort of biological component primarily of the reaction, it eliminates some of that variability. So I think we need to look at what can we do now, and I really admire what FAIR is doing and trying to really address the challenge we have right now with standardizing OFCs and getting the demographics under control, understanding those and the impact in the OFC. But I also think we need to look forward to how will we be measuring sensitivity in the future, and it won't be with an oral food challenge. Of course, that's what we're trying to avoid. It will be more with blood tests and these other measures that are less invasive. And frankly, one of the things we've learned about the oral food challenge is that people don't want to do it, and most people won't. So we need to look at the future, and I think a very near future where we're going to springboard from the oral food challenge, and that's what FAIR is setting up for everyone to be able to do, to a situation where we can assess sensitivity without putting a child and family through an oral food challenge and all of the variability that comes with that, and set that aside and actually get to, I think, a better estimate of what each child's reactivity level is, how much they can ingest safely.
The other thing I would just add, Jason, on that point is that increasingly what we're seeing is the oral food challenge is being used in conjunction with other types of readouts. So you're seeing a tiered approach in which basically people are doing IG testing, they're doing skin prick testing, they're doing basophil activation testing. They're using a panel of different approaches so that they can think about at what point in time do I need to use an oral food challenge, and it may be sort of the final determination that enables you to have a concrete or definitive answer. But it was one of the points that I made in my presentation is that in some cases, individuals don't get an oral food challenge because there's sufficient data based on the clinical history and other readouts that tells you it's highly likely that that individual is food allergic. But to Paul's point, it would be unfortunate that essentially on the basis of all that information, there was a misdiagnosis. And right now, the only way of really truly closing the loop and understanding whether a person is allergic or not is the oral food challenge. So you're talking about perhaps a more data-driven or scientific rationale in terms of when to actually apply the oral food challenge. And aspects related to sensitivity, as Paul's pointed out, I think are critically important. So we're very much keen to think about alternative strategies that can be employed, because it is what we have right now, and this is where the wealth of the historical data is, but we do need to be forward thinking in terms of what will be the alternative in the future. And to remember right now is often with the oral food challenge, The result is binary. You're allergic or you're not. And ultimately, at the end of the day, it would be tremendously beneficial if we could get to a point where we could have a gradation of responsiveness and say individuals have a severe or moderate or a mild form of allergy, because it would, I think, have a direct bearing in terms of the decision as to which therapies an individual might then go on to experience.
Okay. Well, one last question, because I'd be remiss if I didn't indicate this, because there are a couple related questions. COVID-19. So any any reason, I guess, to or any indications that individuals who have COVID-19 are more at risk? In this case, it's from food allergies. And another question that came in was related to, I guess, FAIR's monitoring activity of the population, and is there a conscious decision, I guess, to look at prevalence or incidence in a post-COVID environment?
Yeah, so thank you. So I think the key thing in this area is that when some of the reactions started to emerge or were seen with individuals receiving the COVID vaccine, they were described as being an allergic response. But then I think people sort of rapidly assume, well, does that mean it's a food allergic response? And it turns out that if you go back and look at the database, it's general allergic responses in general, which could be to environmental allergens or to other indications. So people who may have an atopic response. So at this point, from the analysis we've seen, there's no greater risk for an individual, who has food allergies in terms of getting the COVID vaccination. And then the other thing that I think we're just mindful of in this space is just the overall impact of just how the COVID environment has actually influenced, treatment and access to care. So you can imagine that many of the therapies that we have at this day and age actually require patients to go into a clinical site, because if they're receiving oral immunotherapy, it should be monitored by an allergist, because there's always the potential of a response or an allergic response to the actual preparation of allergen that they're receiving. And so that has actually, I think we've seen situations in which it has had a negative impact, in which individuals who are contemplating going on to an oral immunotherapy treatment have decided to forego that until after things have settled down with COVID, because of the whole concern about they had to go into a clinical site in order to receive the therapy. To the degree that in fact we're able to come up with new therapies that could be administered in the home environment, in which it doesn't necessarily always require visiting a clinician's office, that would definitely, I think, benefit the community.
Yeah, and I'll just add to that. Again, I think Bruce really covered that well. One is it's a bit early to tell from the samples that certainly we're collecting if there's a biological association or impact of COVID on allergy and reaction level. I do think the lasting impact will be the greater adoption of telehealth, remote medical care, blood testing. So this, the population actually has already been starting to adopt these technologies well before COVID, but COVID has definitely, the pandemic has definitely accelerated that. So that's fantastic. It's perhaps overdue. So I think that's probably the longer term implication for allergy food management.
Great. Well, with that, once again, Dr. Roberts, Dr. Kearney, thank you very much for sharing your knowledge, your expertise. Very interesting, and very interesting to see what the future state could look like. It's exciting. It's exciting in that space.
Okay.